In our last video, we talked about gradients. So right now, what we want to focus on is the cell membrane, which we see right up here. We've been drawing the cell membrane as a circle. This is also the cell membrane right here, which I will expand on in a second. And this is based off a molecule known as a phospholipid. Understanding phospholipids and the chemical characteristics of them, and as they apply to the plasma membrane, otherwise known as the cell membrane, is super important in understanding the selective permeability of our cells. So our plasma membrane is composed of a phospholipid bilayer. This right here is a phospholipid, which is a very unique molecule in that it has both a polar region, as we see in the purple circle right here, with a positive and a negative, and a nonpolar region displayed by these red tails, if you will. So this is a phospholipid. To be clear, this is polar because it has a positive end and a negative end. It doesn't matter which end is positive or which end is negative, but think of a battery. A battery is polar. It has a positive end and a negative end. And a lot of molecules in our body, specifically the ones based off the atom carbon, are polar molecules. For example, glucose is a polar molecule. Non-polar molecules tend to be, or let me rephrase it, they are steroid-based molecules, lipid-based molecules, and phospholipids like we see right here. Now, I need to restate that. The red component, or the tails of the phospholipid, are non-polar. The head is polar. And that's why this is a very unique molecule, and we refer to these type of molecules that have a polar region and non-polar region as phospholipids. And the cell membrane of our cells, like we see right here, is a phospholipid bilayer. Now, to be clear, I typically draw the cells as a circle like we see right here. That's how I drew it when we talked about chemical gradients in the previous lecture. But this is a phospholipid bilayer right here. This is the cell membrane all the way around the cell. To be clear, as we established last time, outside of the cell we have the extracellular fluid. Inside the cell we have the intracellular fluid, otherwise known as the cytosol. This, what we see right here, is represented by this purple line. And generally speaking, and when I say purple line, this purple line of this cell right here, generally when I'm drawing a cell membrane or plasma membrane of a cell, I'm actually drawing it like this. And the only reason I'm doing that, it's much easier and it's much quicker. But every time I draw a cell like this with the plasma membrane just as one clearly delineated line, please understand that what I'm drawing is this, which is the phospholipid bilayer. So we see these phosphate heads on the outside of the cell, and we also see the phosphate heads on the inside of the cell, and then the tails projecting from those phosphate heads. Hence the name, the phospholipid bilayer, meaning it has two layers. This is one layer on the outside, another layer on this side. To be clear on why it's oriented this way, it's all based off of a polar molecule that is ubiquitous and makes up the great majority of our body, and that is water. Water is a polar molecule. It has a positive region and a negative region. This glass of water, any water in the universe, is composed of H2O or water molecules. And one thing to be very clear, when we say the word water, it could mean one of two things. And let me rephrase it. It does mean one of two things. It is a molecule known as H2O. That is suggesting it has two hydrogen atoms and one oxygen atom. H2O. And if we want to really get a better view of that, this is what the water molecule looks like. There's two hydrogens and one oxygen. And because oxygen is an atom that's extremely electronegative, much more so than the hydrogen atoms, it will pull electrons towards it. So the bonds binding hydrogen to oxygen are known as covalent bonds. And covalent bonds are the sharing of electrons between two atoms. So generally we might draw this as such. Here's two electrons shared between oxygen and hydrogen. But because oxygen is super electronegative, it tends to pull those electrons, and these pink circles, I don't know if they look pink to you, tend to be pulled much closer to the oxygen atom, like so. So while hydrogen and oxygen are sharing these electrons, they are not sharing them equally. That is to say, oxygen pulls those electrons towards itself, and in doing so, imparts partial negative charges on oxygen. And because it's pulling the electrons away from hydrogen, there becomes a partial positive charge on each hydrogen. So we are showing partial positive charges here. And one thing to keep in mind, while I am showing polarity here in this water molecule, this water molecule is a neutral molecule. That is to say, overall it has no charge, but it has partial charges. What's my point in all of this? Is the bulk of the intracellular fluid and the bulk of the extracellular fluid in our body is composed of water, which is a polar molecule. Polar molecules want to hang out with other polar molecules. Non-polar molecules want to hang out with other non-polar molecules. But polar molecules and non-polar molecules do not want to hang out with each other. They don't jibe well with each other. As a result, that's why this phospholipid bilayer is oriented in this fashion. That is to say, we have these polar heads facing the interior of the cell, because the interior of this cell is composed of water, known as intracellular fluid. And by corollary, the outer phosphate heads are facing the extracellular fluid, because the extracellular fluid is composed of water, and water is polar, these phosphate heads are polar. And to be more clear on this, if we were to throw that phospholipid in this glass of water, it would orient as follows without being instructed to do so. That is to say, the polar head is going to happily hang out or float in the water, while these nonpolar tails are going to stick up and get as far away from the water as possible. And once again, that's why the phospholipid bilayer is set up this way. These nonpolar tails in red are trying to face away, not trying, they are successfully facing away from the water that's in the intracellular fluid and the extracellular fluid. This is a phospholipid bilayer. Now, to be clear, this is what allows cells, or specifically the cell membrane, to be selectively permeable. And to be more clear, polar molecules cannot cross the cell membrane or plasma membrane without some sort of channel. A channel allows polar molecules to cross the plasma membrane. Nonpolar molecules can easily sneak through here without a channel. So some sort of lipids or steroid molecules can easily cross the plasma membrane without a channel. Okay, so here's an example of a channel. It doesn't matter what type of channel it is at this point, but just for fun, let's say it's a sodium channel. We've talked about sodium requiring a channel. Polar molecules require channels, and ions, which are charged atoms, also require channels, whether it's a cation, which is a positively charged atom, or an anion. Remember, high concentration of sodium outside of cells and a low concentration of sodium inside of cells. It requires a channel. So here is a channel that would allow sodium to easily move into the cell without coming in contact with these phospholipids, specifically the lipid tail component of the plasma membrane. So this allows sodium to move into the cell, moving down its electrochemical gradient. Channels are composed of proteins that span the entire phospholipid bilayer or cell membrane to allow things like sodium, polar molecules like glucose, among others, to move into the cell. Certainly if we're talking about potassium, potassium would need a channel as well to move out of the cell. I'm not going to draw it just for sake of time, but this is the selectively permeable plasma membrane of a cell. Cells are selectively permeable, totally dependent on what type of channels they have. That is to say, this cell right here is permeable to sodium because it has a sodium channel. Now, most cells are going to have potassium channels as well. Dare I say all cells? I'm not drawing it here, but just for fun, let's assume there's no potassium channels, which I certainly don't have a potassium channel drawn in this image or in this video. So this is selectively permeable only to sodium. Okay, so back to our normal cell membrane, or just a simple cell membrane that doesn't show the phospholipids, but once again, this plasma membrane is a phospholipid bilayer, and we need channels for things to move into cells. So like we drew in the previous image, we can have a channel like this where sodium moves into the cell. We would call this a sodium channel. And there's a number of different types of channels, but I want to be very clear on these channels. Sodium, if it's moving down this channel, and for sake of simplicity, I haven't drawn the brackets around sodium, but please understand that sodium, it's the sodium concentration out here, and it's going to move. Sodium will then move into the cell via this channel. Because sodium is moving into the channel, or let me rephrase this, sodium is utilizing a channel, but it is still moving passively. Sodium is moving down its electrochemical gradient via this channel. And just like sodium, potassium would need a channel, allowing it to leave the cell. Potassium needs a channel because it is charged. To be very clear, once again, sodium and potassium in these cases are just moving down their gradients passively. Because there's a channel, it doesn't mean that energy needs to be used, and I realize we haven't talked about energy yet, and it will be an issue, or it will be a factor in movement of some ions and molecules in and out of cells. But to be clear, this is still diffusion. It is a passive process. Now, this is what we refer to as facilitated diffusion, because it is utilizing a protein channel. Facilitated diffusion utilizes channels or transporters of some sort. To be clear, there is something known as simple diffusion, and these are super small molecules like, say, oxygen or water that can easily move across this plasma membrane, or a lipid or a steroid. Nonpolar molecules do not need channels. So a nonpolar molecule, if it's moving down its gradient, it's going to move through the plasma membrane without a channel. So let's just say oxygen moves into the cell without a channel directly across the phospholipid bilayer. That is simple diffusion. If a molecule or an ion needs a channel, that is facilitated diffusion. The transport is being facilitated or aided by a channel. Okay, so we have simple diffusion, and simple diffusion also works, as does facilitated diffusion, bidirectionally, either into the cell or out of the cell. Now, to be clear on what I just said, I am not suggesting that sodium can diffuse out of the cell, because it cannot. Potassium cannot diffuse into the cell just because the channel is there. This channel allows only, in this case for potassium, only for potassium to move down its gradient, and potassium's gradient is always moving out of this cell. But if we look at something like carbon dioxide, known as CO2, one carbon atom and two oxygen atoms. Carbon dioxide will simply diffuse out of the cell down its gradient from an area of high concentration to an area of lower concentration. And with gases, we're really talking about partial pressures, but to simplify things, we'll just say concentration for now. So there are a number of different channels. These channels I've drawn up here for sodium and for potassium are what are known as leakage channels. That is to say, they are always open, allowing sodium to diffuse into the cell and potassium to diffuse out of the cell. Once again, these are leakage channels. For simple diffusion, no channel is needed. Okay, we have another cell right here. I've drawn it a little bit differently just so I have more space here, and it actually may be more realistic than an actual very symmetric circle. Not that my drawings are symmetric, but I try. Once again, this is a leakage channel. It is always open. This is what's known as a gated channel. Here the gate is closed, and here the gate is open. And specifically, this is what's known as a ligand-gated channel. A ligand, also pronounced as ligand, is a signaling molecule. So for example, a neurotransmitter or hormones are ligands. They are signaling molecules. And there's other molecules in the body other than hormones and neurotransmitters that function as ligands or as signaling molecules. If the ligand binds to this channel at this receptor right here, it's going to open up that gate. These right here, which are really the same channel, are ligand-gated channels. That is to say, a ligand, which is a signaling molecule, binds to the channel and opens up the channel. No ligand is bound to this channel here, so the channel is closed. Here, the ligand is bound to it, so the channel is open. So to be very clear, the ligand is never what passes through the channel. So for example, this could be a sodium-gated channel where if this channel opens, then sodium in the extracellular fluid is going to diffuse passively into the cell. But that's only going to happen when the ligand binds to it. Now, certainly, if a cell membrane has leakage channels, sodium can come in whenever it wants. But when we're talking about gated channels, this is now a way for the cell to regulate when and what enters the cell. So this is an example of a ligand-gated channel. Now this right here is what we refer to as a voltage-gated channel. A voltage-gated channel is a channel that is activated when there is a change in voltage in the cell. So we've talked about the interior of cells being negative and the exterior being positive. If that voltage in the interior of the cell changes, generally becoming more positive, it will activate voltage-gated channels. I also like to refer to these as voltage-sensitive channels because these proteins are sensitive to the change in voltage. So if the cell is at rest and it is not depolarized, if the cell is at rest, it will have a positive charge on the outside and a negative charge on the inside. If for some reason this polarity changes to positive, trust me, we haven't talked about this, but if it does change, and we'll explain how that does happen, that change in voltage from negative to positive will open that gate. Now, to be clear, this is what's known as a voltage-gated channel, but that's still an incomplete description of the channel because we need to know what is permitted to move through that channel. So once again, like this one is a ligand-gated sodium channel, this could be a voltage-gated sodium channel. And to be clear, it could be a voltage-gated potassium channel. Really, the way I've drawn it, it would be a voltage-gated sodium channel. So once again, sodium, there being a lot of sodium outside the cell, if there is a change in voltage in this cell, it's going to allow the influx of sodium into that cell via the voltage gated or voltage sensitive channel. So we have ligand gated channels, we have voltage gated channels. There's another channel type called a mechanically gated channel, and when there's some sort of physical change or pressure or manipulation of a cell, it tends to open, or it will open up, a mechanically gated channel. So a mechanically gated sodium channel or a mechanically gated potassium channel. So in short, we have leakage channels, ligand gated channels, voltage gated channels, and once again, we also have mechanically gated channels. So as far as gated channels, we have ligand gated channels, voltage gated channels, and mechanically gated channels. I want to stress that if there is a channel being utilized, it is still moving the entity through the channel passively down its chemical gradient or electrochemical gradient. We will see glucose channels where a glucose channel opens and glucose passively moves into or even out of a cell.